Have you ever really thought about what happens to the water after it goes down your drain? It's carrying all sorts of organic waste, and if we just let that flow straight into our rivers, well, it would be a huge environmental mess. But it doesn't, all thanks to this hidden world of tiny microorganisms that we've actually put to work cleaning up after us. So let's dive into this incredible process of biological wastewater treatment and see exactly how we turn polluted water back into a healthy, thriving part of our environment. Here's the game plan for this explainer. First, we need to understand the problem we're trying to solve, what pollution actually does to a river. Then, we'll look at the brilliant biological solution we've come up with. We'll get into the details of two major technologies, something called trickling filters and another called activated sludge. After that, we'll see how operators keep these living systems in balance. And finally, we'll wrap it all up with the key takeaways. All right, so to really get why the solution is so clever, we have to start with the problem itself. What exactly is the environmental impact that makes wastewater treatment so absolutely essential? It all boils down to one single vital element: oxygen. Let's ask a simple question. What really happens moment by moment when a pipe carrying untreated wastewater, which is full of organic stuff, dumps into a perfectly clean, healthy river? The answer is more than just the river gets dirty. It actually kicks off this fascinating and destructive chain reaction that environmental engineers call the oxygen sag curve. This diagram here shows that chain reaction perfectly. This is the oxygen sag curve. Let's walk through it. As you can see, it tracks the amount of dissolved oxygen in the river as you move downstream from where the pollution enters. In the clean zone, way before the pollution, oxygen levels are high, and you've got healthy fish like trout. But then, bam, the wastewater hits. This introduces a ton of organic matter, which we measure as biochemical oxygen demand, or BOD for short. That's the food. Now we're in the decomposition zone. The bacteria that naturally live in the river start having a feast on all this new food. But to do that, they need to breathe, and they consume the dissolved oxygen right out of the water. You can see the oxygen level just plummet. As it drops, only tougher fish can survive. If the pollution is really bad, you hit the septic zone. Here, the oxygen is almost totally gone. Fish die off, and you start to get those nasty, rotten egg smells. But the river keeps flowing. Eventually, the bacteria run out of food, the river starts pulling in new oxygen from the air, and we enter the recovery zone. Oxygen levels start to climb back up, and life slowly returns, bringing us back to a clean zone far downstream. And this graph really drives the point home. The depth of that oxygen sag is directly tied to how much pollution you dump in. A little bit of BOD creates a small dip, and the river recovers pretty fast. A medium load creates a much bigger, more damaging sag. But a high BOD load, like what you'd get from a whole city's untreated wastewater, can crash the oxygen level to almost zero, creating a massive, miles-long dead zone. So, the entire goal of wastewater treatment is to get rid of that BOD before it ever reaches the river, to prevent this sag from ever happening in the first place. So, if the problem is a biological process, bacteria eating waste and using up oxygen, it makes sense that the solution would also be biological. Instead of letting this chaos happen in a river, we decided to build a controlled, super efficient environment to do the exact same thing inside a treatment plant. And that's the whole idea behind what we call secondary treatment. After the first step, primary treatment, which just lets heavy solids settle out, the water still has a lot of dissolved organic gunk in it, that high BOD we were talking about. Biological treatment uses a huge, concentrated army of microorganisms to eat that dissolved organic matter. And it's incredibly effective, removing 90 to 95% of the BOD and a lot of harmful pathogens along with it. We're basically taking that decomposition zone from the river and building a supercharged version of it. Now, there are two main ways to get this done, and it all depends on the type of bacteria you use. First, you have aerobic treatment. This uses bacteria that need oxygen, just like us. It's a really fast process, and the byproducts are clean, mostly just carbon dioxide and water. You have to pump air in, so it costs more in energy, but it's super efficient. This is the workhorse for most city treatment plants. The other way is anaerobic treatment, using bacteria that work in zero oxygen environments. This is a much slower process, kind of like fermentation, and it produces things like methane gas. It's cheaper to run, but not as good at cleaning the water itself, so it's usually used for treating the leftover solid sludge. For our purposes, we're going to focus on the aerobic methods. This right here is the simple recipe for aerobic treatment. On the left, you've got your ingredients, the organics from the wastewater, which is the food. You've got the bacteria, who are going to eat it, and you pump in oxygen for them to breathe. Through their metabolism, they turn all that into what's on the right. More bacteria, which we call new cells, and harmless stuff like carbon dioxide and water. The really key idea here is that we're turning a dissolved pollution problem into a solid problem, bacterial cells that we can physically remove from the water later on. Now, even within aerobic treatment, engineers have two main strategies, and it's all about how you manage the bacteria. In attached growth systems, we give the bacteria a surface to live on, like rocks or plastic, where they form a slimy layer called a biofilm. Then we just let the wastewater trickle over them. The classic example is a trickling filter. In suspended growth systems, we just let the bacteria float around and mix freely right in the wastewater. The most famous example of this is the activated sludge process. Let's break each of these. Okay, let's kick things off with that first major technology, the attached growth method. It's called a trickling filter, and the name pretty much tells you exactly what it does. This schematic shows you the entire setup. You've got this big, round tank filled with some kind of porous media. It used to be just rocks, but now it's usually special plastic. Wastewater gets pumped up to the middle and sprayed over the top with these rotating arms, kind of like a giant farm sprinkler. As that water trickles down through all the media, it makes contact with the biofilm. That's the slimy layer of microorganisms growing on the media surface. Those microbes eat the pollutants out of the water. Air naturally flows up through the gaps in the media, giving them the oxygen they need, and at the bottom, the clean water is collected. Every now and then, the biofilm gets too thick, and a layer will just slough off and get washed away with the water to a settling tank where it can be removed. Now, the media you choose is super important for how well the filter works. What you're looking for is a material with a really high specific surface area. That just means tons of little nooks and crannies for the biofilm to grow on in a small space. Of course, it's got to be cheap and tough enough to last for decades. And maybe most importantly, it needs to have high porosity, which means lots of empty space. This is crucial for preventing the filter from getting clogged up and for allowing plenty of air to circulate for the microbes to breathe. The old-school trickling filters were called standard rate. You'd apply the water very slowly. They were cheap to run, but they took up a huge amount of land, which made them expensive to build. The big leap forward was the invention of the high-rate filter. These can handle a much higher flow of water, which means the whole system can be a lot smaller. And the secret sauce that makes it all work is a clever trick called recirculation. And that brings us to the key feature of high-rate filters, recirculation, which you can see in this flow diagram. We use a pump to take some of the water that's already been treated and send it right back to the beginning to be mixed with the incoming wastewater. This gives us a few huge advantages. First, it dilutes the strong incoming waste so the microorganisms don't get shocked. It also adds a little dissolved oxygen back into the water, and it keeps the filter media wet all the time, which helps keep a healthy biofilm growing. So how does an engineer actually design one of these things? Well, it's not just guesswork, it's a very logical step-by-step process. As you can see here, you start by figuring out how much water to recirculate, then you calculate the total amount of pollution you need to remove, and from there, you can figure out the size, the volume and area of the filter itself. Let's just quickly peek at the thinking behind a couple of the key calculations. So the very first thing to figure out is how much water needs to be recirculated. The formula might look a little intimidating, but the idea is simple. It's just a balancing act. On the left side, you're calculating the total amount of pollution coming in. That's from the main flow plus the little bit that's in the recycled water. And you balance that against the target pollution level you want for the water being sprinkled on the filter. You solve for that QR, the recirculation flow, and that tells you exactly how fast to run the pump to keep the system happy. Okay, once you know your flows, the next big question is, how big does the filter need to be? Well, first, an engineer calculates the total weight of pollution in kilograms that needs to be removed every single day. Then, they look up a value for the filter media they chose. You can think of it as the media's cleaning power, how many kilos of BOD it can remove per cubic meter of media. You just divide the total pollution by the media's cleaning power, as you see in the formula, and that tells you the total volume of media you need to buy. Simple as that. All right, let's shift gears now and talk about the other major technology, activated sludge. If a trickling filter is like a fixed apartment building for bacteria, activated sludge is more like a giant, constantly moving party. This is what we call a suspended growth process. This diagram shows you the whole process. Water from primary treatment enters a big aeration tank. Inside, it's mixed with this thick, brown, bacterial-filled goop called activated sludge, and air is constantly bubbled through it. This whole mixture is called the mixed liquor. After a few hours in there, the mixture flows into a secondary clarifier. This is just a quiet tank where the heavy sludge can settle to the bottom by gravity. The clean, treated water then flows off the top. Now here's the magic trick. Most of that sludge that settled is pumped right back to the beginning of the aeration tank. That's the return activated sludge, or RAS. This keeps a really high concentration of hungry bacteria in the tank at all times.
And to keep the population from growing out of control, a small amount, the waste-activated sludge, or WAS, is removed from the system each day.
Now, pumping all that oxygen into the aeration tank uses a ton of energy. It's one of the biggest costs for a treatment plant. And there are basically two ways to do it. You can use bubble or diffused air systems. These have a grid of diffusers on the bottom of the tank that release tiny bubbles of air, kind of like a fish tank bubbler, but on a massive scale. They're pretty energy efficient, especially in deep tanks, but they cost more to install. The other method is surface aeration. This uses big mechanical mixers, like giant egg beaters, to churn the surface of the water and mix air in from the atmosphere. These are cheaper to install and easier to maintain, but they're not quite as energy efficient. A really crucial thing that makes this whole process work is something called flocculation. See, a single bacterium is way too tiny and light to ever settle out of the water on its own. But in the aeration tank, they start producing these natural, sticky slime layers. This makes them all clump together with other particles into these bigger, heavier clusters we call flocks. These flocks are what become heavy enough to actually settle out by gravity in that next tank, and that's how we separate our clean water from our colony of helpful microbes. And this is what it actually looks like under a microscope. It's not just a random blob of bacteria. It's a whole, complex, living ecosystem. You can see the main blobby structure of the flock, but if you look closely, you can see other organisms too. You've got protozoa, which are actually good guys, because they swim around eating stray bacteria, helping to make the final water even clearer. You also see these long, stringy filamentous bacteria, which kind of act like a backbone that holds the whole flock together. Keeping this little ecosystem healthy and balanced is the key to good treatment. And to keep that ecosystem healthy, you have to feed it a balanced diet. This table shows you what a typical bacterial cell is made of. About half of its dry weight is carbon, which it gets from the waste. But look at the other important ingredients. About 12% is nitrogen, and about 2% is phosphorus. These are essential nutrients, like vitamins for the bacteria, that they need to build proteins and DNA. Most regular domestic wastewater has plenty of N and P, but some industrial waste might be missing them. In that case, the plant operator would actually have to add these nutrients to the water to keep the bacteria healthy. So we've designed these amazing engineered ecosystems, but how do you actually run them day to day? Well, controlling a living process is part science, part art, but it all revolves around one central idea. You have to balance the amount of food coming in with the number of microorganisms you have to eat it. And that brings us to what is easily the single most important control number in the entire activated sludge process, the food-to-microorganism ratio, or FM ratio. And it's exactly what it sounds like. The food is the amount of BOD coming into the aeration tank every day. The microorganisms is the total weight of active bacteria we have living in that tank. By carefully managing this one ratio, operators can steer the health and performance of the entire plant. But how on earth do you weigh microorganisms? You can't just count them. Instead, there's a standard lab test. An operator takes a sample of the mixed liquor from the tank. They filter it, dry it, and weigh what's left. That gives them the mixed liquor suspended solids, or MLSS. But that's everything: living bacteria, dead bacteria, sand, grit. To find just the active microbes, they take those dried solids and burn them in a super hot furnace. This incinerates all the organic stuff. All that's left behind is inorganic ash. The weight that was lost during burning is called the mixed liquor volatile suspended solids, or MLVSS, and that is our best measurement for the mass of active microorganisms. So, with those two key pieces of information, the incoming food, which is the BOD, and the mass of microorganisms in the tank, the MLVSS, operators can finally calculate the F/M ratio. You divide the total kilograms of food per day by the total kilograms of microbes in the tank, and you get a single number that tells you the health of your system. Most plants try to keep this ratio in a Goldilocks zone, somewhere between 0.2 and 0.6. So what happens if that ratio gets out of whack? Well, if the F/M ratio is too high, it means there's way too much food for the number of microbes you have. We call this young sludge. The bacteria are basically overfed and growing too fast, so they don't produce that sticky slime needed for good flocculation. This means they won't settle well in the clarifier, and your final effluent will be cloudy and turbid. A classic visual sign is a thick white foam on the aeration tank. To fix this, an operator would waste less sludge, letting the microorganism population grow bigger. And the opposite problem is a low FM ratio, which we call old sludge. This is a starvation situation, not enough food to go around for a huge population of microbes. In this case, the bacteria form these really dense, compact flocks that settle incredibly well, almost too well.
They can be so brittle that tiny little pieces, straggler flock, break off and escape in the final effluent. To fix this, an operator would do the opposite. They'd increase the sludge wasting rate to remove more microorganisms from the system and bring that FM ratio back up into the target range.
Wow, okay, we have covered a lot of ground here, from the problems happening out in our rivers all the way to the nitty-gritty details of controlling these amazing engineered ecosystems. So let's just take a quick moment to tie it all together and recap the most important points. First, remember the oxygen sag curve. It's that perfect visual that shows us exactly why we need to treat our wastewater in the first place. Second, the solution is biological treatment. We're just using microbes to eat the BOD in a controlled box instead of in the river. We looked at the two main ways to do this: trickling filters, which is an attached growth system with a fixed biofilm, and activated sludge, a suspended growth system where the microbial flocks are mixed right into the water. And finally, we learned that for activated sludge, that food-to-microorganism, or FM ratio, is the absolute key. It's the main control dial operators use to keep the whole living system balanced and working perfectly. The ideas we've talked about today are the foundation of wastewater treatment all over the world. But the challenges we face are always changing. Today, our wastewater has all sorts of complex new contaminants, things like microplastics, pharmaceuticals, and chemicals from personal care products that these systems were never designed to handle. And that leaves us with a really important question for the future. How can we take these principles and build the next generation of microbial ecosystems, ones that are smart enough and powerful enough to tackle the pollutants of tomorrow?